My name is Shohei Nakamukai from Japan. Let me start my presentation. So, natural products are utilized in the various fields such as fragrance and medicine. So, many bacterial natural products have been isolated from the marine organisms. So, especially marine sponges are known to contain many bacterial natural products. And they have unique and complicated structure compared to those on land. So, among them, peptides containing non-proteinogenic amino acids, PNPA, is a characteristic compound found in marine sponges. PNPA can be categorized into two types: so non-ribosomal peptides (NRPs) and ribosomal synthesized and post-translationally modified peptides (RIPPs). Both peptides contain several non-proteinogenic amino acids, which is thought to be crucial to their biological activities. For example, daptomycin, which is an NRP used as a medicine, contains some D amino acids, ornithine, and some modified amino acids, and even ethyl bond and fatty acid chains. And polyphenol B, which is a large RIPPs, forms the helical structures by the interaction of the polarity of the D amino acid. And so this molecule acts as a channel for the divalent ion across the cell membrane and exhibiting potent cytotoxicity. So the main method to identify the secondary metabolites is the analysis of MS-MS spectra. So to quickly and correctly analyze identify the secondary metabolites, the comprehensive database is essential for comparison with measured data. However, a problem with marine natural products, including PNPA, is the scarcity of MS-MS data in the database. So in the past studies, so the rates of structured identified MS-MS analysis is 18.5% in plants and 6.7% in marine sponges. So therefore, it is difficult to identify the marine natural products by the MS-MS analysis. So in recent years, so some study reported the utilization of retention times of LCMS for the compound identification. However, even the same compounds, the retention time can vary depending on the measurement conditions, such as columns, solvents, and flow rates. So therefore, Bash et al. Focus on the retention order, which is the order the compound eluted from the LCMS column. So if the reverse phase condition is the same, the retention order does not change significantly. So it is possible to compare with a different dataset. So in the past study, they succeeded to predict the retention order of a small molecule by using the machine learning rank SVM and achieve the to predict the prediction accuracy with around 0.9. Therefore, so in this study, we conducted the retention order prediction of PNPA. There are several benefits in the prediction of retention order. For example, we can train the compound without MS-MS data. And also, we can use the data from the measured under different conditions. However, a problem with PNPA retention order prediction. There is a significant problem. This is the scarcity of the training data of PNPA in the database. So as a solution, we employ the new approach that we train the amino acid portion of PNPA from the peptidomics data. And we train the modified portion of PNPA from the small molecule data. And using the prediction model, by using the prediction model, we conducted the retention order prediction of PNPA and for the reference compounds. And after that, for the evaluation of retention prediction model, we compare the predicted retention order and actual retention order. So as a data set, we use the Predret database. It contains small molecules. And the peptidomics dataset, it contains the peptides composed of proteinogenic amino acids. And we also prepare the 22 reference compounds of small molecule and 9 PNPA standard. These test data were measured under the same conditions. And so this is a prediction model. So we employ the RNA-QSBMM. And first, we made the compound pair in the same dataset and represented them into the fingerprints. Fingerprint is a bit strings indicating the substructure exists or not in the compound structure. So the fingerprint expressed with 0 and 1 is binary fingerprint, and expressed with counted substructure is called counting fingerprint. So by using this fingerprint as input data and using the retention order as output data, we conducted the training for all possible compound pairs. We tried the optimization of fingerprints. So light blue indicates that only peptidomics data was used, and light green indicates that only small molecules was used. And light blue and light green indicates that both was used. And we also compare the difference between when using binary fingerprints and when using counting fingerprints. And furthermore, we compare the two types of fingerprint. Max fingerprint, which is a typical fingerprint representation method, and mixed fingerprint. This is a mixture of 4 types of fingerprint. And this graph is results. So the prediction accuracy tends to be higher when counting fingerprint was used. And it also be higher when mixed fingerprint was used. However, in some conditions, the red one, the results were reversed. So in these all red conditions, the both counting fingerprint and mixed fingerprint were used. So therefore, we calculated the variance inflation factor, VIF, which is the indicator of the correlation between variables. And as a result, so the prediction, VIF values of mixed counting fingerprints tend to be higher than other conditions. Therefore, so we thought that one of the reasons of the low prediction accuracy of mixed counting fingerprints is the multicollinearity. So principal component analysis, PCA, is one of the methods to suppress the multicollinearity. Therefore, we conducted the PCA to the fingerprints and obtained the principal component fingerprint, PCF. By using this PCF, we performed the same training, and this is the result. So red one indicates improved prediction accuracy. So as you can see, in many conditions, so the prediction accuracy was improved by using the PCF, and especially so in mix counting in PCF, the prediction accuracy was significantly improved. And in the average, the prediction accuracy of mix counting incomplete was the highest. Therefore, so we chose the mix counting PCF as the input variables for the rest of this study. We conducted the variable reduction to improve the prediction accuracy. We systematically selected the input variables from the smaller component which have higher contribution, smaller component numbers which have higher contributions. As a result, we could obtain the highest prediction accuracy of 0.904 when using 200 of variables with peptidomics and small molecule dataset was used. So we success to predict the retention order of PNPA with high prediction accuracy of over 0.9. Next, so we investigated the prediction accuracy for each PNPA. So except for 2 PNPAs, so most PNPA had high prediction accuracy of over 0.9. Among them, the 3 compounds, vasopressin, alpha-amanitin, epsilon B, were completely predicted. So vasopressin is composed of all proteinogenic amino acids. And epsilon B, this is not peptide, but the structure of this is partially derived from amino acids. Therefore, we saw that the higher prediction accuracy of these 2 compounds is due to that the structure of these compounds, their structure is close to those of peptides and those of small molecules, respectively, in the training data set. Yeah, okay. So, next, to investigate the structure affecting principal component, we analyze PC1 and PC2. And so, we identify the top 5 variables which highly significantly influence on the PC1 and PC2 respectively. So for PC1, there are many structures associated with amide bonds. And for PC2, structures associated with 6-member rings and oxygen atom attached to ring structure were contained mainly, indicating that the hydroxy group attached to ring structure or sugar moiety significantly influenced on PC2. So, in summary, so we succeeded to predict the retention order of PNPA, whose training data was scarce in the database by utilizing the peptidomics data and small molecule dataset, and achieved prediction accuracy of over 0.9. And in the future work, we aim to improve the prediction accuracy by enriching the training data and carefully selecting the reference compounds. And we hope to incorporate the retention of the prediction into compound identification in the future. Thank you for listening.
Thank you. So now we take questions and comments from the audience. Okay, please. Lim-sun.
In your approach, I missed some parts, but if I understood it correctly, the training is based on the PC principal components. Is that correct?
Yes.
Yeah, but the principal component is the deconvoluting the variation in your data sets. Then for future data, their variation, especially the batch effect one, may not be part of the original PCA space. So then your model will have this gap, right? Because the new data would have variation that doesn't belong to the old space. So when you project them using the same coefficient, would that be a problem?
Sorry, so what's the question, sorry?
So I was saying that your model was trained based on the principal components. Yes, but the principal component is derived from your current training data sets. So it only captures the variations in the current data sets. In your future data, their variations may not be part of, will not be similar to what you have in your training data. So when you project, I assume you are going to apply the same coefficient to derive the PC values, or project the new data into the old principal component space. But the new data has its own variation that is orthogonal to any of the existing PC, or then what will happen?
So new data is not affected by the principal
It will be,
analysis.
because your mass spectrometry has its batch effects. So the new data will have variation that doesn't belong to, or direction of variation that is not present in the original training data sets. But your principal components only capture the direction of variations that is present in the original data, but not new directions.
So yeah, so that is probably one of the a reason of the low prediction accuracy in the test data. Thank you.
Okay.
So into sorry. So into compounds. So this is the worst prediction accuracy was obtained. So probably in the training data, so this the training data couldn't capture the feature of these two compounds. So
Okay, so please
that is
discuss after the session.
yeah, thank you.
Okay.